Genetic improvement of crop plants. Crop improvement is vital for global food security and sustainability. By developing high-yielding, disease-resistant, and climate-resilient crops, we can increase food production, reduce crop losses, and enhance nutritional content. This leads to improved food availability, economic benefits for farmers, and a more sustainable agricultural system, ultimately contributing to a food-secure future. Selection and hybridization are fundamental techniques in conventional crop improvement. Selection involves identifying and propagating plants with desirable traits from a genetically diverse population. This process, whether natural or artificial, is used to enhance characteristics such as yield, disease resistance, drought tolerance, and quality traits. Hybridization, on the other hand, refers to the deliberate crossing of genetically diverse parents to combine favorable traits from both into a new, superior progeny. Traditional breeding, while effective in developing improved crop varieties, has several limitations. It is often time-consuming and labor-intensive, requiring multiple generations to stabilize desired traits. The process relies heavily on visible traits, phenotypes, which can be influenced by environmental conditions, making selection less precise. Moreover, it offers limited control over specific genes, leading to the possibility of transferring unwanted traits along with desirable ones. Traditional methods are also less effective for complex traits governed by multiple genes, such as drought tolerance or yield. Additionally, it depends on the availability of genetic diversity within sexually compatible species, restricting the scope of improvement. Inducing mutations involves exposing seeds or plant tissues to radiation or chemicals like EMS to create random genetic changes. These mutations can produce new traits such as higher yield, improved nutrition, or resistance to pests and diseases. Chromosome doubling is used to restore fertility in hybrids. Example, in triticale, a hybrid of wheat and rye. This process improves adaptability and stress tolerance in the crop. Molecular markers linked to desirable traits enable precise identification of plants carrying target genes. They allow early selection without waiting for plants to mature, saving time and resources. This technology accelerates the breeding process by quickly screening large populations. As a result, it improves efficiency and increases the chances of developing superior crop varieties. Genomic selection uses genome-wide markers to capture genetic variation across the entire genome. It predicts the breeding value of plants based on genetic data rather than solely on field performance. This approach enables early and accurate selection of superior breeding lines. It accelerates crop improvement, reducing breeding cycles and boosting genetic gains. Gene transfer from other organisms introduces specific genes into crops to improve traits like pest resistance or nutrition. Examples: Bt cotton, golden. These innovations enhance yield, reduce pesticide use, and address nutritional deficiencies in agriculture. Gene editing in agriculture precisely modifies an organism's DNA to improve traits without introducing foreign genes. Techniques like CRISPR-Cas9 allow targeted changes for disease resistance, yield improvement, or stress tolerance. It enables faster crop improvement compared to traditional breeding methods. This approach supports sustainable farming by reducing chemical inputs and enhancing climate resilience. Rapid generation advancement uses controlled environments to shorten crop growth cycles. By manipulating light, temperature, and other conditions, multiple generations can be grown in a year. This speeds up breeding programs and accelerates the development of improved crop varieties. Genomics, transcriptomics, proteomics, and metabolomics provide multi-level insights into plant biology. Together, they reveal how genes, RNA, proteins, and metabolites influence plant traits. This integrated approach enables precise crop improvement and stress adaptation strategies. Bioinformatics applies computational tools to analyze large-scale biological data in agriculture. It supports trait prediction through methods like genome-wide association studies, GWAS, and QTL mapping. These approaches help identify genetic regions linked to desirable crop traits for targeted breeding. Climate-resilient crops are developed to withstand stresses like drought, heat, and floods. AI and machine learning accelerate breeding by predicting performance under diverse conditions. Participatory breeding involves farmers directly in variety selection, ensuring locally adapted solutions. Genetic improvement is crucial for securing the world's food supply. Combine traditional breeding with modern tools ensures faster, more resilient crop development. Thanks for the time. If you like this video, please subscribe channel.